Hi everyone, my name is Shawn Small, and I'm a senior studying psychology and biology at UMass Amherst. Thank you for joining me at the Massachusetts Undergraduate Research Conference. Today, I'm going to share with you my research on behaviors performed by the eastern carpenter bee, Xylocopa virginica. Specifically, we'll be looking at behaviors performed at the entrance to the eastern carpenter bee's nest, and we'll be looking at its vibroacoustic behaviors performed inside of a nesting gallery. Vibroacoustics encompasses vibration and sound production. Historically, behavioral observations have been recorded with a pen and paper. My data collection instrument automates the observation process. This device is not intended to replace pen and paper observations. Rather, it's a demonstration of what you can do with simple hardware and open source software. Together, these tools can be used to create a custom data collection instrument tailored towards studying the behavior of a focal species. This is an ethogram covering the behaviors performed by the eastern carpenter bee. An ethogram is a record of all known behaviors performed by a species. Here you can see on the left is a list of the behaviors performed by eastern carpenter bees, followed by a description of what these behaviors entail, and concluding with frequencies of these behavioral performances. One of the primary goals of my thesis research is to produce a more comprehensive ethogram of the eastern carpenter bee's behavior. Let's briefly discuss the species' nesting behavior and its development. One food source for the eastern carpenter bee is pollen, and a female bee will go out and collect granules of pollen and bring them back to its nesting gallery. The female bee then compacts this pollen into a ball, called a pollen mass. Once the female has made a pollen mass, it places it inside of what's called a brood chamber. The female then lays an egg on top of the pollen mass and seals the combination inside of the chamber with wood shavings, which you can see here. The female does this repeatedly until it's laid all of its eggs for the season. As a univoltine species, carpenter bees raise one brood of offspring annually. While adult bees overwinter in hibernation, the young juvenile bees start developing inside of these brood chambers and eat their pollen masses, getting ready to emerge come springtime. In the picture on the left, you can see carpenter bees that have developed inside of their brood chambers, with their wood shaving partitions separating each chamber. Female carpenter bees demonstrate facultatively social nesting. Facultative sociality is a plastic behavioral phenotype, which means there's flexibility in how the behavior is performed. Some female carpenter bees will build a nest on their own, and they're considered to be living in solitary. Other carpenter bees will share a wooden structure and build their nests amongst other carpenter bees. These carpenter bees are semi-social. The full range from semi-social to solitary nesting is the facultatively social component of the species' nesting behavior. Whether a female carpenter bee chooses to nest in solitary or in a semi-social setting depends on space availability. Male carpenter bees are territorial. Once they establish a territory, they will fly at and buzz at any intruder to the territory. If these male bees also engage in female defense polygyny, which means a male will defend any breeding females inside of its territory. So if, for example, a male has a territory that contains a nesting gallery, the male will defend all of the females from intruders that live in that nesting gallery. If you're curious about how to distinguish female carpenter bees from male carpenter bees, here are a few quick tips. If you look at female carpenter bees, you'll see they have large mandibles, which are essentially the carpenter bee's jaws. Female carpenter bees use these large mandibles to bore their tunnels in wooden structures. Males of this species do not bore tunnels in wood. If you look at the male carpenter bees, they have this spot on their head, and that spot is a clear indication of a male carpenter bee. Regarding the species' behavior, I asked 3 questions. First, I want to understand how often the species perform their behaviors. Does a carpenter bee perform one behavior more frequently than the other? I also want to understand what the species' circadian activity pattern is. So I'm looking for when the species is most active during the day and when the species is least active during the day in a 24-hour period. Lastly, I'm investigating what sound-producing behaviors are performed inside of the nesting gallery. Once again, Vibroacoustics covers vibration and sound production. To observe the bees, I built artificial nests. Here you can see the entry hole to one of the artificial nests. Connected to that hole is a 40 centimeter long tunnel that the bees will live inside of. On the right, you can see one of the nests erected upon a 2x6 board. After building the artificial nests, it was time to extract carpenter bees from their natural nest. With a combination of wood chisels and saws, I opened up these carpenter bee tunnels, and with tweezers I extracted them one by one from their nests. Next, it was time to move the carpenter bees into the artificial nesting galleries. So I again picked up the carpenter bees with tweezers, and I placed them into their new nesting galleries, one by one, in the same order in which I removed them from their natural nests. After that, it was time to set up the data collection instrument. This data collection instrument is built around a Raspberry Pi Zero, which is a single board computer. A single board computer can do all the things that your computer can do, just maybe not quite so quickly. Attached to the Raspberry Pi Zero is an ArduCam day-night camera. This camera records in the visible light spectrum during the day and in infrared at night. At any point in time when motion is detected at the entrance to a nesting gallery, the camera will save the footage locally on the Raspberry Pi's microSD card and remotely in a shared network folder. Next, you can see I'm attaching the uninterruptible power supply, which is the pink Pi Juice board. The uninterruptible power supply comes with a battery, and together they regulate device electrical current flow. If the power were to go out, the battery will turn on and prevent the device from losing power, even in the event of a mains power outage. This way, data collection continues even if there's power loss. Here you can see I'm adding audio cables to the device. These audio cables are connected to transducers that are attached to the outside of one of the artificial nests. The transducers provide audio input to the Raspberry Pi, and that's how I record sounds produced within the nesting galleries. Here, I'm using hot glue to hold everything in place. Next, it's time to add the thermistor. The thermistor is a contact thermometer. In this case, I'll be using the thermistor to record daily air temperatures. Now, it's time to attach the ArduCam to the enclosure cover. Now, once sealed, this enclosure is rated to be watertight and dust resistant, so the data collection instrument will operate without concerns of weather damage. For data collection, the instrument is running automated interval sampling, meaning that any and all activity observed outside of the nesting gallery via the ArduCam camera will be recorded and saved by the Raspberry Pi. All sounds produced within the gallery will be recorded with the transducers. Once again, I'm using the thermistor to record daily air temperature, and I will use ArduCam footage to measure the photo period. Here, the photo period represents the amount of time when there's incident light upon a nesting gallery. For data analyses, I'm first going to compare the date and time stamps on the datums. By comparing the date and time stamps, I can figure out when the species is most active and when the species is least active. I'm going to count the cumulative performances of each recorded behavior to understand how often eastern carpenter bees perform each of their behaviors. And then I'm going to perform sound source localization on the vibroacoustic data. Sound source localization is a method for identifying where sound comes from. So if you look at the person in the center of this figure, they have two ears. And sound is localized on three axes: the elevation, distance, and azimuth axes. With these three axes and a central point, you can figure out where sound is coming from. If you look at the figure on the right, you can see the blue and red ears. If we think of each of those ears as microphones, and we see that a sound wave is arriving at them, we see that sound reaches each microphone at a different time. Using those differences in time, plus these three axes, we can figure out where sound comes from. Now, to date, as of the time of recording this video, no carpenter bees have emerged from their artificial nesting galleries yet. If you tune back into my YouTube channel in about a month's time, I'll share with you another video covering my data collection, my analyses, my results, and my conclusions. I'd like to thank Dr. Stephen E. Brewer, my mentor and leader of the Open Science Instrumentation and Data Collection Thesis Seminar. I also want to thank my LSIDC peers, Emmett, Andrea, Shawn, and Andrew. Dr. Brewer and my peers alike have provided me with invaluable feedback throughout the course of my research. I next want to thank Xander Crowley. Xander ordered all of the parts that I used to build my data collection instrument. I also want to thank Michael Davidson. Michael Davidson provided me with a carpenter bee infested board, from which I extracted carpenter bees to add to my artificial nests. I now want to thank the Commonwealth Honors College for providing me with funding to conduct this research. And I want to thank my anonymous reviewers who, without their feedback, I wouldn't have received my grant from the Commonwealth Honors College. Next, I want to thank the ICONS faculty members and my peers in the 8th color cohort. ICONS faculty members and peers have provided me with support covering this research. If you'd like to learn more about my research, feel free to check out any of these links. The top link goes to my research website, which provides an in-depth detail on my thesis research. Next, if you want to see the code that runs the data collection instrument, feel free to check it out on my GitHub page. If you're looking for weekly updates on my research, feel free to check out what I've posted on Public Lab, which covers my research progress from last fall to today. And to view that video in a month's time, covering my results from this study, feel free to check out my YouTube channel. Once again, I want to thank you for joining me at the Massachusetts Undergraduate Research Conference this year. And I hope you enjoyed my presentation on my thesis research covering the behaviors performed by the eastern carpenter bee, Xylocopa virginica.